I'd like to introduce our next speaker, who is a person I've known for years. He's been involved as a director and as a president of the society, and is now our outgoing past president, as well as chairman of the scholarship committee. He is a associate professor at the University of Hartford. He currently is an associate editor for the Journal of Aquatic Plant Management and the Journal of Plant Ecology. And he's going to be speaking today on efficacy of Clearcast and Procellacor EC on European frogbit. Ben, you're on.
Hey, thank you, Greg. I would also mention that Mark Hyman from CIPRO is a presenter for this project and also collaborator for this project as well. So hi again. Thank you for keep staying here. Try to listening to last 3 presentations. So I'll try to keep short as possible as I can. So I like our incoming present, Dr. Cassie McLean mentioned in the morning that we live in the invasions, right? Biological invasion is happening around us, and it becomes more and more frequent. As you can see here in the graph, showing in the Great Lakes, right? We have about 180 introduced species. There are more and more coming in the recent years. You know, here are some zebra mussels, a spiny fish hook, water flea, bloody shrimp. You know, these are the plants. You know, we care. Our society care about this. Water chestnut, Eurasian milfoils. So unfortunately, all these species come in mostly due to us, our activities. For example, this one showing you the ship ballast dump and load in the last. Not last, but in the 14 months, showing you they're going everywhere, right? Whenever they go to a port, they take the load and then go to another one, they dump it. So when they do this, they transport a lot of invasive species to different places. So you can see here in the Great Lakes, again, showing you some of the places are hotspot of introductions, right? Here in Lake St. Clair, we have like 10 introductions. The famous zebra mussels are founding over here, starting from here and then spreading around. So because our activities, and we have this hotspot, right? Here just showing you looking at the Great Lakes region. If we look at it globally, you can see here in the oceans, we do have a lot of intensive hotspot of invasions as well. For United States, looking at the West Coast and the East Coast are very serious. Here you can see very dark colors, right? The dark colors means that we have intensive invasions. So all these invasions cause negative impacts, right? So that's why we're concerned. Not only in the oceans, not in the waters, on the land, the same thing, right? Looking at here, again, in the United States, we have California, Florida, Texas, we have a lot of invasions here. Northeast, generally, it's not so good either, right? So we have those colors showing you a lot of invasions. It's great that we gather together, do the studies, do the management. And I thank all of you very much for doing the hard work. Climate change is also a factor affecting the invasions. Studies have shown that they are facilitating invasion of the new species to the new areas. So that caused the problem. One of the example I show you today is a floating plant, European frogbit. Right, so I did a study on this plant about 12 years ago, 14 years ago, looking at the invasions in the Finger Lakes regions. By studying here, showing that the global warming actually promotes the plant growth. So it's very impossible for the plants to spread around. And also it didn't show any effects of the nutrients. So it doesn't matter if it's allotropic lakes or eutrophic lakes, it doesn't matter. So they can be there, they can survive and establish and spread and have negative impacts. So what is European froglet? It's a floating plant, and it has this white flowers with the yellow inside. It's pretty nice. So it started from Eurasia, introduced from the Switzerland to Ottawa Botanic Gardens, and then escaped. Go through the St. Lawrence River, go to United States in the 1970s, right? So they can spread by means of water, birds, boats, and human activities. So this is the one found in New York State, that the pond has a lot of European froglet. So should we worry about this plant? This one study show that they produce from stolons and water buds, right? For the stolons, as you can see, that they can expand from one to another, from 1 to 2 to 3. So it grows rapidly. And they have about 100 buds per plant. So that also contribute to their growth and fast growth spreading to the areas. And they can form very dense mats. For example, this one showing you 500 plants per square meters in Oneida Lake. And because they can reduce the dissolved oxygen, right? So that dissolved oxygen is important for invertebrates, for fish. That's because it's reduced, that's going to have negative impacts for those organisms. And also it shows that it has a positive impact on surface invertebrates. I think this is understandable because they're plants, because those invertebrates like plants, so there are more of them, but they don't have any impacts on the benthic macroinvertebrates. However, it has a strong negative impact on submerged macrophytes, especially those native plants, right? So we want to conserve those native plants, but European froglet presence will have negative impacts. Kathleen showed you that it reduced common bladderwort to like only 10%, right? So, and also our study showed that it's changes the structure and composition of native plants as well. So because they have huge impacts, and then we need to be concerned. And also because it's spreading fast. Here's showing you that spreading from Ottawa regions and then expand to more and more and more areas, right? More and more areas. So in 2007, we found that in Oneida Lake, in Cayuga County, and also I think in Audubon of the southern Chautauqua County as well. So here showing you a map from the USGS that it's limited in the northeast region around the Great Lakes area, right? But we should be concerned because they're more and more spreading. And then here you can see in Long Island regions, they have this species as well. So we want to be careful with them, and then we want to manage them to avoid them to spread into more and more areas, right? So there are some of the studies showing some control methods. Some of them show more effective than others. Biological control using snails did not show any impacts. Okay, so in the field we find, in those European froglet, we have find a bunch of snails actually attached on the leaves and stems. But when we did the experiment, it did not show that snails have any significant impact on controlling this plant. And also we did tries on shading with the shed cloth and also hand pulling. They can remove European froglet effectively. However, shading has some negative impacts on macroinvertebrates and native plants. and henna pulling involves a lot of labors, and right, so you need to go there frequently. First we did try once a month. It did not show any impacts or effect, but then we changed to frequency to 1 to 2 weeks, and then it shows control effectiveness. So it's going to involve a lot of labor cost. What about other control methods? So this is what we're thinking. You know, we look at the literature, paraquats, anthosols, diquat, they have shown effectiveness in controlling European frogpit. Those studies were done like in 1960s and 70s. Our goal for this project is to look at the two herbicides, Clearcast and Procercor EC, on controlling these plants. So that's why we did the experiment. Actually, we did two independent experiments. One is at my university, University of Hartford, in the greenhouse, and also the other one is in CPRO Research and Technology Campus in North Carolina. And we got the plants from the Bashkir Wildlife Refuge in New York. Thanks to Nicole White from DEC, sent us those plants, and then we separate them in grow them in 1 to 4 weeks, culture them, and then do a random test in 5 groups. These are 5 groups. One is using as a control group. There's no herbicide treatment at all, so we call it a UTC group. Next is the Clearcast at the 32 ounce per acre. So that's a C32, we label it. That's you can show that in the figures. Clearcast 64, we double mount the concentration of the Clearcast. Then we use the Procercor EC at 10 PDU. Then we tried another group, is the Clearcast 32 and the Procercor 10 PDU, a combination of 2 chemicals. So these are the 5 groups we tested looking at the herbicide effectiveness on European frogpit. We measured them. We targeted the concentration as 20 gallon per acre. We measured dry mass, leaf damage, and also green canopies. Right, so looking at the green canopy, what's the coverage at the end of the experiment. So the experiment lasted 4 weeks. Here showing the experiment in UHART experiments, looking at the different buckets. So each bucket has 3 replicates. So we have those 5 groups: treatment group, UTC, C32, 64, P10, and a combination of Clearcast and Procercor. So have the 2 plantlets at the beginning. We apply the chemicals, and then after 24 hours, flush all those away, and then replaced with the new river water. So here showing you at the end, right? So these are the control group, and you can see them are relatively healthy. This is C32 group. The 2 buckets showing completely death. This one showing some of the greens. This one, because it's the first application of the herbicide, there's an arrow for when my student did the application, so they actually have a lot of leftovers in the spray. So they didn't have the complete amount there. So when we did the statistics analysis, this one is actually excluded. Then C64, the double concentration, you can see basically they killed all of them. And this one showing you P10. P10, some original leaves are dead, but then you can see there's new growth, new growth in those plants. The combination of those 2 chemicals, they are completely dead at the end. So if we look at the graph showing very similar thing, I just talked about looking at the picture. The dry mass, there's those 5 groups, right? UTC, C32, C64, and the P10, C32, and the P10. These 3 groups, C32, C64, and also combination of the Clearcast and the Procercor, they pretty much killed everything, right? And the Procercor did not have a significant impacts, right? It compared to the control group, it has a lower concentrate, a lower biomass, but you can see the amount still a lot of them there, also shown in the picture as well. By looking at the plant final survival rate, this is showing you a very similar pattern. If you look at the damage of the leaves from the beginning week 1 to week 4, that showing you similar patterns. The C32, C64, and also the combination of those 2 chemicals, that they did not show very significant impacts at the end, but you can see at the beginning there's slightly differences, okay? And then Procercor, and this is the control. Control basically means that they did not damage, most of them survived. All right, I move on to hand over to Mark, talk about the results from CPRO.
Hey everyone, Ben, can you hear me?
Yes.
Excellent. First off, hello to everyone. I'm Mark Heilman, Director of Aquatic Technology for CPRO. I'd first like to thank Northeast APMS and the board for all their efforts with the virtual meeting. Not ideal, we'll push for 2023 again in person. Also would like to thank Ben for his collaboration and partnership on this trial that looks at a couple of CPRO's technologies for selective control of invasive aquatic plants. And in this case, European frogbit, an emerging threat, it appears, in a variety of northern US waters. One other thing I'd like to add is there is a collaborative associated with European frogbit, and reach out to me, or you can query it on the website. A variety of scientists and managers from around the country have gathered to compile information about ecology and management strategies for this plant. So reach out to folks involved with the collaborative if you're looking for broader information. And so with that, I'll switch gears into the duplicate trial that we were able to implement here at the CPRO Research and Technology Campus, which I'm actually sitting in the conference room of today versus my normal confines in the Indiana office. And I wanted to run through first a visual look. The trial methods are very similar. We did treat plants that were probably a little bit further along and a little bit larger mesocosm settings, about a 57-gallon tank, I believe, no, 57 liters, excuse me. And what you see here is after the 4-week study, the condition of plants in these various treatments. Untreated controls, for the most part, quite healthy. We did have one replicate tank that didn't behave itself, but you see dense growth in the other untreated plant replicates. With treatments of Clearcast at 32 fluid ounces to the acre or 64 fluid ounces to the acre, we see essentially close to complete control. Procercor is probably a fairly interesting one to talk about, applying EC at 10 PDU per acre. Just for volume context, that's 32 fluid ounces to the acre. And you see maybe some injury, as Ben was talking about. There are some effects. We see some leaf curling, some epinastic growth early after treatment, maybe a little bit of die off of those first leaves treated. But what we see is that that effect is pretty transitory, and we see fast regrowth and generally strong recovery of the plant. So European frogbit is not one that we would recommend for Procercor alone and control the species. However, we did, as Ben referenced, look at a combination treatment with Clearcast at 32 fluid ounces with 10 PDU of the EC per acre. And that did look quite good. Maybe a slight slowdown in initial symptoms of control versus Clearcast only, but basically caught up quickly with equivalent results. And so this supports that the two herbicides can be used in combination if you were looking at a mixed invasive plant population with European frogbit and another species more sensitive to Procelacor. And I'll reference that again at the end. Ben, can you advance? So this is just dry biomass. If you looked at the data that Ben presented earlier, you might see more biomass versus the study at University of Hartford, but the trends of the error bars are basically the same. We had some variance in our untreated controls, but we did see 3 treatments that were statistically significant. Clearcast alone at 32 or 64 fluid ounces to the acre, or Procelacor at 10 PDU EC in combination with Clearcast at 32 fluid ounces. So all 3 of those treatments showed significant effects statistically relative to the untreated control. Procelacor alone did not. And again, both in biomass and also another metric of injury, what we did from a methods perspective is use the Canopio app. This analyzes imagery for green coverage of plants. It's developed by Oklahoma State, I believe, a number of years back, and has been used in a few different published studies. We used that technique to take the photos similar to what we showed earlier and look at the green cover associated with each of the European frogbit tank populations in the study and characterize percent green cover using that technique. And you can see that the untreated controls were averaging about 70% overall green coverage. Some of those replicates obviously higher than that at the end of the study versus the Clearcast rates, and also the Procelacor combination with Clearcast showed very little green coverage, mostly open water and brown and necrotic plants at 1 month post-application. Procelacor alone, as indicated earlier, did not show much longer-term injury, so by 4 weeks was equivalent in green coverage to the untreated controls. Next slide, Ben. So in conclusion, with the results, we did see both in biomass and green coverage through Canopio that the Clearcast at 32 or 64 fluid ounces to the acre, as well as the combination with Procelacor, all those treatments showed significant impacts on biomass and green coverage of the healthy plants. Visual leaf damage was observed within 3 days after application for those treatments. Procelacor alone, just to kind of get into those symptoms, did show again some curling of leaves, twisting, some brittleness or the shattering effects that we see with Procelacor on some plants, breaking at growth nodes or meristematic regions. And that was observed through about 1 week after application, but that effect was temporary. We saw healthier growth continue and then bounce back to basically the same level as untreated controls by the end of the trial. So in conclusion, the data support that 32 fluid ounces of Clearcast or the higher rate at 64 fluid ounces both would be sufficient alone to provide good control. A combination of Clearcast at the 32 fluid ounce rate with Procelacor EC at 10 PDU EC per acre foot, or per acre, excuse me, it's an important distinction. Both provided, all those provided effective control within 4 weeks with a good transition in control between 2 and 3 weeks. Procelacor EC alone has low efficacy, and that's why it's not labeled for that species. Next slide. Did want to reference that a catalyst for much of this work was questions coming from those involved with the European Frogbit Collaborative, and also a particular project managed by New York State Department of Environmental Conservation. Their efforts on the Peconic River with Ludwigia control, invasive water primrose, it happened that surveys found European frogbit mixed in with the Ludwigia, and therefore there was an interest to find a treatment strategy or determine if a treatment strategy that involved Clearcast and also Persellacor that was planned for the primrose, if that would be effective on European frogbit. And so we appreciate the collaboration with DEC to generate some data that was supportive of their efforts on the Peconic River, and that those appear to be aligned in initial research or initial demonstration efforts have shown that both frogbit and Ludwigia are being controlled in the field at that site. Next slide. Just a few other things. A use advantage of these treatments, both Clearcast and Persellacor have very few water use restrictions relative to drinking, swimming, etc. at these use rates. With the combination, we do have a very temporary short irrigation precaution associated with the Persellacor side of the treatment, just half to 1 day after application with the foliar treatment. If larger scale management would be considered where there's sites with good water retention, and if the standards is perhaps to try to fully remove as much European frogbit as possible, in water applications of Clearcast and possibly also Gallium, another related ALS herbicide, the active ingredient penoxylum. Both those may also be considered and could be areas for additional research in the future. These could be used alone or as systemic herbicide follow-ups to contact treatment. Operationally, in the Midwest, for example, flumioxazin is shown to be pretty active as a contact treatment on European frogbit. And I think I'm done, Ben.
Okay, thanks, Mark. So, well, our future work, I think when we think about the herbicide treatment, the one thing we need to think about is how they're going to affect the native plants, right? Especially those rare native plants, for example, the alternated flowered water milfoil, like a water marigold. So those species are very rare. Are they going to be impacted by herbicide treatment? That's maybe one of the directions of the future research. We are focused on the herbicide treatment, but one thing I would remind all of you and all of us, I think you all know that early detections, preventions, early detection and rapid response still is important for control invasive species, including European frogbit, right? So this graph showing you the cost when you have a lot of cost once they're introduced and they are established, then it's really hard to get rid of them. But if we find them, we prevent and early detect and rapid response, that's probably have a low cost and have a good control success of controlling invasive species. Again, monitoring is also very important. I think a lot of you have done great work in monitoring invasive species and monitoring other native plants. At the end, I would like to acknowledge Chase Kilgore and Ben Willis at the CPRO for managing the experiment at the CPRO, and Jing Yu at the NYU for assisting the experiment and the data analysis. And again, I thank Dr. Cassie McGlynn at the DEC for discussions and start of the project, Nicole White from DEC, and Lauren Henderson at the Capital Region PRISM for collecting and shipping the samples for us to do the test. With that, we would be happy to answer any of the questions.
Okay, can you hear me?
Yes.
Oh, good. Okay, yeah, we have some questions here. It was a great discussion. First one is, are calcium concentrations a large determining factor on what water bodies frogbit can invade?
The calcium concentration, we didn't test any calcium concentration in this project. I'm not so sure about the calcium concentrations.
Okay. And just one correction from Nicole White. She said it was Lindsay Yoder at LH Prism, not Lauren Henderson. So,
Oh,
correction.
sorry about it.
Okay. All right. With that, I do thank you guys very much, and we'll look forward to your future work, and we'll move on to our next presentation. Thank you again.
Thank you.